So as Barnabas said, I'm going to be on the course from the side of neuroscience, and we've been working with Andy for a few months together, and it's an interesting journey for me because it's a strongly interdisciplinary project. We should, to understand, really, we should be knowledgeable in both all of art and technology and science, and specifically in my case, neuroscience. And I think in order to navigate this, the confluence of these three disciplines, we need to use triangulation. So, there are many people here and in the team who are knowledgeable in art and technology, and I have to kind of try to provide a frame of reference from the side of neuroscience. And I'm going to talk about the technique and so about electric brain activity, which is also called EEG. And I will explain how we measure EEG and what does it kind of mean in a biological manner. And I will try to explain some visualizations and kind of art pieces based on this background knowledge. So when I was a PhD student, I studied attention using this method, and of course we make data visualizations even as scientists, but these are just to summarize and show the data really well. So, and Andrea, for example, she's, for example, I've seen her work as like a consumer, or so to say, of art with the band Muse, which is they made a nice, beautiful audiovisual live act and installation, and these are between science and technology and art and technology, and we should explore the territory in between. So. One thing I want to talk about is this is a visualization made by the Gedzalei lab in the United States. It's called the Class Brain, and it visualizes the electric activity of the brain as a whole. And another piece is a collaboration between György Buzsáki's lab, a very famous Hungarian neuroscientist, and visual artists. So both are about the electric activity of the brain, which is almost exactly 100 years old. So the first EEG recording was made by Hans Berger in Jena at the 6th of July, so it was last Friday, the 100 years anniversary. So interestingly, his motivation was to understand remote sensing and these psychic powers. So, but he, in the meantime, he described alpha waves at the top and beta waves, and basically we even today use very similar methods to study the brain's electric activity non-invasively, so from the outside. So I trained as a biologist, and this is what a brain looks from the inside. These are microscopic images from rodent brains. And we've learned as a biologist that there are like billions of cells in there. We learned about their development, their shapes, and their molecular biology, and how they communicate with themselves. But I think it was hard to answer the question that how can it be that just a homogeneous bunch of nerve cells or brain cells can give rise to the mind and cognition. And another thing that was, as a biologist in training, fascinated me, that how is it possible that we can record meaningful signals from outside the brain? It's like, it felt like listening to a football match from outside the stadium, so it didn't quite click. So let's look at the first question. So let's evoke the image of a cartoon neuron from a textbook. So real neurons, of course, don't look like this, but of course they have soma, which is basically the input of the cell, and they have an axon, which is the output. And suppose we have another cell, and that this cell sends an input to this specific cell we are talking about. And we can put a little electrode in the cell in question and measure its so-called membrane potential. You can think of it as a kind of like an activity level of the cell. And if it receives an input, there's a little bump of activity level, which in this case doesn't reach that threshold with dashed line. So the cell doesn't produce an output for now. Similarly, we can imagine another cell which could also send an input, and it also doesn't produce any output. But if we activate through both cells simultaneously, then we can these activity bumps can add up, and they cross this threshold, and the neuron produces its output, which is called spikes and firing. So spikes can travel really fast to other parts of the body or the nervous system. So these are the communication signals, and those activity patterns are just local computations. And I'm saying computations because we've just implemented a simple logical operation in a cartoon neural circuit. So the output only activates if both inputs are active. And this is also called an integrated fire neural model. So information scientists in the past decades have implemented artificial neural networks using simple computational units conceptually similar to what I described. And we know by now that these networks are capable of doing very complex tasks. So what do real neurons look like? We can see that they have a very intricate shape. So they have the soma is not like a blurb, but it has intricate processes called dendrites. And if I zoom in on that segment, each of these little outgrowths on the dendrite are receiving ends for another neuron's input. So because of this geometric complexity, basically these real brain cells have vastly more computational power than just a simple logical operation that I described on the previous slide. So probably you would need a few hundred cartoon neurons at least to model what a single brain cell does. So what I'm getting at is that if even the artificial neural networks, which are relatively, say, simple compared to the brain, can compute like complex operations with the power of the complexity of the brain cells, basically cognition could, in theory, be an emergent property of the brain as a complex neural system. So the second question is how we are able to even record something outside of the brain. So to answer this question, we have to look at the brain from the outside, because we are lucky, because this outermost layer of the brain that we see is the so-called cerebral cortex. You can also see this gray band in the cross-section. And so this means that the cells are close to the surface where we can actually place our sensors, which is like good. So another thing is that the cells that are concerned with a specific function, like recognizing a face, are not randomly scattered around the brain, because it would be not really ecological to send signals to a long distance. So these are clustered in functional brain areas. For example, there's an area in the temporal cortex to process faces, or another area for printed words. And when I described this logical circuit, there was an important prerequisite that the two inputs have to be synchronized. And if we imagine the neural circuits from these cartoon neurons in these brain areas, that these neurons have to do something together, they have to have some method to coordinate temporally their activity to be able to communicate as a neural ensemble that perform a computation. And a simplification that I made on that side, that I suggested that the activity of the neurons is like 0 before receiving an input, but this is not true. Actually, this activity level is fluctuating, and there are not so good times to give an input to evoke output, and at some other times the same intensity of input can evoke an output response. So if these fluctuations would be random, that would be not too good, because the cells would fall in and out of the population and they could not work together. So because of this, neurons can produce rhythmic activity level or excitability fluctuations, which are called neural oscillations, and they have a way to do this in a synchronized manner. So neurons that work together are basically orchestrated by neural oscillations. And let's look at real neurons again. So the neurons in the cerebral cortex have like an elongated shape, and neurons with an elongated shape produce a larger electric field that we can measure. You can think of it as a little antenna, illustrated by the plus and minus sign. And also, these cells are not randomly aligned in the brain tissue, but they are perpendicular to the surface of the brain. And this also means that if there's these antennas basically can add up to one big antenna. It can be sort of a antenna array if the activity of these neurons is synchronous. And I have shown in the previous slide that it indeed, mostly it happens that they fire and work synchronously. So we have cells at the outermost layer of the brain, which are elongated and ordered nicely in like this column-like structure. They are organized into functional clusters, and their activity is coordinated temporally. And by all these being true, we can measure EEG from outside the brain. And you can probably see that while I was answering two separate questions, I arrived at some points where the two questions converged. Because I was telling about a functional thing that neurons have to synchronize themselves as a population to be able to do computations. And also, it was a prerequisite for the summation of the signals to be strong enough to be measured from outside the brain. So I think, so there's an overlap between mechanisms that enable cognition to emerge and between mechanisms that allow the biophysical emergence of the EEG signal. So because of this, the neuroelectric signals that are meaningful with respect to the function of the brain can be measured from outside the brain. Okay, so let's look at this glass brain again. But beforehand, so let's suppose we have a spot where the neurons are synchronized and they are producing this oscillation that is measurable from the outside. And maybe there's another population that also produces the same pattern. You can think of that it also means that if these populations are connected by axons, the output projections of the neurons, then these two brain areas can communicate. And this is what is visualized here, because the so we can see the cortex, and the glowing colors represent oscillations of different frequencies that are synchronized locally in a brain area. And the little running lights reflect these connections inferred based on this model that similar oscillations imply communication between brain areas. And the other installation, it is not very closely using neural data, but it's rather illustrating a nice concept. Okay, can I? Okay. So the oscillations in the brain work like in a hierarchic structure. So slower oscillations can coordinate lower frequency oscillations and so on. So it basically creates a neural syntax that is a bit like natural language syntax, that like letters constitute words and words constitute sentences. It similarly happens in the brain with oscillations nested into each other. Also a bit like music. And after this, I think I might have sold EEG to you. So I've been telling that it's a great tool. It's a great tool indeed. But I have to tell you that you probably think that those are EEG electrodes. But I think it's better to think of them as just electrodes, because they don't measure brain activity, they measure electric activity. Electric activity is produced by muscles, eye movements, eye blinks, but of course also by the brain. But as the font size illustrates, these other signals coming from other parts of the body can be very strong. And there's even an activity that you can't read, I think, from your seats. So I zoom it in for you. So this is an activity related to individual events. So it's just a word of caution about the instantaneousness of EEG, which is sometimes not really true. To characterize EEG responses to individual external and internal events, you really have to collect and aggregate a large amount of data sometimes. And also scientists and also artists need to somehow assure that if they want to work with brain signals, they have to try to avoid the unwanted other biological signals, which can be, of course, interesting, but not brain signals. And also the brain source location of EEG signals is not straightforward to tell, so it can be quite ambiguous where the signal exactly comes from. So I put up this triangle again because if I'm a scientist, I have to plan a study in a way that I deal with these issues. And if you are an artist and you are working with concepts for neuroscience, but not directly the data, then it's maybe, I guess it's not so important. But if you want really a data-driven piece, then it's also important to deal with the same questions that we struggle with as neuroscientists. So this is a challenge for us during the course in the coming week. So thank you for your attention, and why does it fade?